So I've been in the field of gene editing for almost 4 decades. And in the early days I was described as a rebel, and now I'm seen more as an elder statesman and sometimes a pioneer. I think I like the rebel designation a bit better. And after years of elucidating the molecular mechanism of gene editing in human cells at various universities, I had the opportunity to join ChristianaCare to create the Gene Editing Institute that would focus on translating our basic science discoveries, what we call benchtop science, into applications that could eventually be used at the bedside. That decision revolutionized the way my team and I think about science and even how to study gene editing. So we learned primarily from being at ChristianaCare and embracing their values that the patient needs to be considered in the development of any innovative therapies. And that guided our thinking. And so we came through the process and ended up in a place where we would say our work is a patient-first approach to gene editing.
I mean, when we started, we were doing pure mechanistic research. How are these things happening, on the biological level? But then when we had an application for it all, and being embedded in the cancer center at the time, it really put the emphasis on the patient.
When you're in the lab and you're a lab rat and you're so used to just looking at your cells and you say, oh, these are cancer cells, it's different when you're in the elevator with the people that are suffering that disease. And you press the number for that person and they say thank you, you're like, that's the person that I'm trying to help. To take that science and give it a face, I think that has really been what the patient-first approach has given us, is just putting a sense of humanity in the type of work that we do.
So I'm a firm believer in maintaining a competitive edge in the field of cancer research, particularly in the area of CRISPR and gene editing. And while we've made some significant progress, and I think GI has contributed a great deal to the field, we have to continue to innovate and move forward because frankly, what got you here won't keep you here.
Our biggest research accomplishment is really being true to the science. We are very methodical with how we do our science, and while we have major publications, journals such as the CRISPR Journal, Nature Communications, I think what really drives our research is the fact that we always follow the data itself.
The difference with what we do is that it's a completely new paradigm of medicine. It's not like other medicines that we understand. This is truly going after the genetic level.
We are working on a gene involved in resistance to anti-cancer drugs. NRF2 is a transcription factor that is involved in various different downstream pathways responsible for oxidative stress, stress response, metabolism. And this gene in particular does cause cancer cells to become resistant to chemotherapy and various other forms of therapy. We realize that this gene is causing patients to really suffer and not be able to respond to treatment. And so we've been making it our mission to find a way to use CRISPR to be able to give these patients a second chance to be responsive to chemotherapy or their standard of care.
So Decoder is an analytical tool that we use to make sense of our gene editing reactions. We developed it based on a need we had for that type of analysis in the work we were doing, and we were fortunate enough to have a high school aged intern, Rohan Kanchana, join our lab, and he was able to use his programming skills to come together with us to develop this program. It has grown legs. It's now used broadly in the field, both in academia as well as in industry, in major research centers, hospitals, all over the world.
So one of the most important things about this patient-first approach is really to try to engage younger folks within the community to help them understand the power and the implications of gene editing. And it was really that led to the development of CRISPR in a box.
So another researcher and I were looking at repair mechanisms of DNA. So we wanted to see what the system could and couldn't do, and then along the way we found that it actually could be used to teach students, either high school or college, about the work that we do here at the Gene Editing Institute. So the Learning Lab is a concept we started back in 2021 when I became program manager, and it was just an idea. And then in 2022 to 2023 is when we really piloted the first learning lab, where students could come on site for a 3-hour field trip experience and experience CRISPR in a box, an education kit that students get to walk through the same steps that I did when I was developing the kit. When students come to Learning Lab, they actually get to experience the skills that we do in the lab itself. So they are learning hands-on skills, and they're learning it all before college. Micropipetting is something I never did until I got into college, but now that they're learning it earlier, it's going to set them up for college or for whatever they get into right after high school.
We were quite successful in getting about 1,000 kids in our first year to attend our Learning Lab. What happened was that it suddenly started to gain some traction at the national level and the international level. In fact, there are programs with the Learning Lab using CRISPR in a box being taught in Delaware and in Texas and in Hawaii and even in Guam. That's been really rewarding for me to see, because we've had a good idea that was grabbed by a talented group of people. The development of the Learning Lab that has gone nationally has really been an astounding measure of the outstanding work that's been done in GEI. So these two missions, translational science and community outreach, have become embedded in our principles at GEI, our values. Most of the efforts on the research side have been intensely focused on developing CRISPR as an anti-cancer drug. This led to the development of an intellectual property portfolio. We have many issued patents and many pending patents. I think we actually were the first to be issued a patent at ChristianaCare.
We do patent globally. The reason for that is there are large markets across the globe, and the ones we're interested in therapeutics, particularly in lung cancers or cancers of the head and neck. There's high incidence rates of those occurring in certain populations, for example, in Japan.
So as we progressed with our translational research group, it started to become apparent that we had developed a path and certainly an approach to treating head and neck cancer, lung cancer, and perhaps several other indications. That journey is quite expensive. And so in 2022, ChristianaCare and GEI spun out the first company ever from Christiana, known as CRISPR Therapeutics. I've had the pleasure of being the founder of that company with folks from ChristianaCare. And over the course of the next 2 years, we have developed a clinical path, a clinical plan, and engaged some top physicians at ChristianaCare and around the country, frankly, to develop the journey toward FDA approval. It doesn't work by itself. You need people who've done this before, and certainly over the last year, we've been delighted to have senior folks from Spark Therapeutics, from IMVAX, and from other companies in the Philadelphia region who have already had this kind of success to join CRISPR and take it across the finish line. So folks ask me about where does GEI go in the next 10 years.
It's definitely moving towards the therapeutic side of things. We're finally seeing that happening.
There's new assays being developed, there's new technology being developed, new CRISPR tools. So it's really changing every couple of days, every couple of months.
I hope that we've been able to enhance people's quality of life, or at least enhance the quality of the end of their life.
We really hope that eventually we can expand even further and impact more students, because we've seen how it changes their trajectories for college or even their careers.
I believe if we maintain that uncompromising dedication and unrelenting pursuit of excellence, I have no doubt that our talented new leadership team will continue to innovate strategies for gene editing in cancer and even in rare diseases, and as always, exceeding expectations.